Okay. What's the time? Okay, guys. Are you here? He said good night. Yeah, welcome. Yes, everybody. I'm just coming back. Hello, good evening. How many people do we have here? 25 people. Okay, I guess the other people are coming, but I'm not waiting for them. What happened? What time did I even say before that we are supposed to start? 8 o'clock, right? 8. Unfortunately, I finished my class around, I'm supposed to finish by 9:30, actually. I forgot I finished Monday by 9:30. So, but I had to hurry up so that I can finish around 8. I got here like 10 minutes away. So, don't worry. We go as much as we can, all right? Yesterday, I had a review and we started from cell. So today, I'll start backwards from muscles, bones, backwards so that anywhere we are tired, we can stop. Then you can easily go watch the other video, all right, where we discuss cell. But in the other video that was yesterday, we didn't discuss bones, muscles, genetics. Instead, we did cell, macromolecules. And also chapter 1 and thereabouts. All right. Okay. Nice to have you guys here once again. So I'm going to share my screen and we start immediately because we don't have much time. So I'll share my screen and we start ASAP. Yeah, you guys are welcome. Sorry that I didn't come on time. Sorry. No, this is neuro. I was supposed to even finish the bone video today, but I was so busy. So I was not able to post it. Yes, so today we are starting with muscles. And I'm going to explain before hold on. I'm going to explain before we start answering the questions, all right? As usual. And if you have any question or any concern, please do well to tell me during the course of the discourse. Sorry. Let me put my phone and not disturb. Just a minute, please. I need to be sure that there will be no disturbance. Okay, so that works. All right. Yeah, we're up to 30 here. Okay, 37. Good. So I will be very fast. All right. I'm not teaching this night. I'm just trying to run through this thing as soon as possible. Then I start throwing question right, left, and center. You know how I do it now normally. All right. So today we are starting with chapter 11. That's muscular tissue. All right. That's really important that you understand the foundation of muscular tissue. The muscular tissue, we have 3 types. We have skeletal, we have cardiac, we have smooth muscle. All right. They have high level of organization. All right. Now, these are the characteristics of muscle: responsiveness, that's excitability, ability to stretch, to understand chemicals around it, and also both electrical changes as well. Now, conductivity, all right, contractility, shortening and lengthening, extensibility, elasticity. That's ability for it to return to its original resting length after being stretched. These are the universal characteristics of muscle, which each and every one of you has to know. All right. Now, starting with skeletal muscle, what do you need to know about skeletal muscle? It is voluntary and it is striated. That's the most important thing. If you look at it, this is the wavy striation. And what is the cause of this striation? The alternating light and dark, light and dark band. What am I even saying? Okay, fine. Please, if I'm too fast, just tell me, indicate it on this thing so that I can slow down, because right now I'm so hyper. All right. So as we said, skeletal muscles are voluntary. Skeletal muscles contain a muscle fiber. This muscle fiber is a cell. I mentioned initially, if you watch my muscle, hello, welcome. If you watch my muscle video, I mentioned specifically that muscle fiber is a cell of its own. You know how you think about a cell, that's how you think about muscle fiber. All right, that's what you need to know. Another name for it is myofiber. All right, so you need to know that. If you put that in your mind, then you are good to go. Now, if you look at this is a prototype. I hope I'm not lagging. No, I don't think I'm lagging. So if you're looking at this image, you need to know what endo, peri, and epimysium is. All right, now, assuming this is a room, this thing now is a room filled with students. The wall of that room is what? Is epimysium. The wall of that room is epimysium. And epimysium is a connective tissue that surrounds the entire muscle tissue. So it surrounds the entire muscle tissue. Now, if you come into that place filled with students, what do you see? You see a group of students. Oh, maybe African students, all of them will be in one place. Latin students will be in one place. Jamaican students will be in one place. American students will be in one place. Do you understand? Just group of students, group. So each of those groups is called what? A fascicle. Mind you, each of those groups is called what? A fascicle. You get it? And each fascicle is tied. So each group is tied by what? Perimysium. So perimysium is a connective tissue that ties each group inside a muscle tissue, all tied by the wall, which is what? Epimysium. Now, if you now enter one of the group, maybe the African group, for example, you bring one student. You understand? You see that one student is what? Muscle fiber. All right. And if you look at the students will be wearing clothes. So these clothes will be serving as what? Something that is wrapping the student. That is what? Endomysium. So you must know endomysium, perimysium, and epimysium, what they are covering. And also you must know that muscle fibers makes up of or rather, let me start from the biggest to the smallest. Muscle tissue made up of muscle fascicles. All right. Now, the muscle fascicles is made up of what? Muscle fiber. So if you look at this diagram, each of these big circle is muscle fascicle. In each of the muscle fascicle, you will see what? Muscle fiber. All right. So if you bring out this one fiber, you bring out one fiber out. All right. If you bring out one fiber out, one fiber, you will now see what? Inside the one fiber, you will see organelles. So those organelles, I said, assume that they are called what? That the organelles are called myofibrils. All right, myofibrils. One of the myofibrils is myofiber. All right, then the myofiber is made up of what? The actin filament, the myosin filament, and all those other things. But what you need to learn from this particular slide is what ties them up. I'm taking time here because this is very important. You are going to be tested on this, definitely. All right, so they can give you a diagram and point to some of the structures. You need to know what you are looking at. You need to know if you are looking at the entire muscle tissue. You need to know if you are looking at the fascicle. You need to know if you are looking at one particular muscle fiber. So don't mistake muscle fiber to a fascicle. Don't mistake a fascicle to a muscle tissue. Okay. So now let's go to a particular muscle fiber. A particular muscle fiber, I said consider a muscle fiber as a cell. All right. Cell has what? Cell wall, plasma membrane, nucleus, blah, blah, blah. This, that. The same thing a muscle fiber has. The cell wall or the plasma membrane is known as what? Sarcolemma. All right. Then it has the cytoplasmic, the internal environment, which is cytoplasm. We call it what? Sarcoplasm. And it has the bundles that we call what? Myofibrils. All right. They just think of the myofibrils as what? Just think of it as organelles, all right? Then we also have the glycogen, we have the myoglobin. The myoglobin is the red pigment, all right, that helps for oxygen activities. While the glycogen gives it energy. Remember, it needs oxygen and energy for it to be able to make movements. So that's why they are very, very dense in a muscle fiber, all right? Now, we have cells like satellite cells, myoblasts, but I'm jumping them. Now, this is the sarcoplasmic reticulum. Very, very, very important. All right. Why is it important? They will ask you, what is the function of the sarcoplasmic reticulum? It is to store calcium. It's a calcium reservoir. And that sarcoplasmic reticulum has two wives beside it, or two sisters. All right. We call them sisters. Okay. Let's not say wife. Let's say sisters. Let's keep it godly. Okay. Those sisters are called what? Terminal cysteines. All right. They are at the two sides. Now, in the middle is the T tubule. You see my hand? So if I'm the T tubule, here is one sister, here is another sister. All right. So you need to understand this structure. Now they may give you this diagram and tell you to point. They point to different things. Anything that is red like this is mitochondria. We know that. All right. Now this place that is light is the I band. This place that is dark is the A band. I will still show you more about this. This is the nucleus. This is the sarcolemma. And if you look here, you see the T. This one in the middle, I think that's purple. You understand? Is the T. But beside it, I guess that's blue. My eyes is not working very well. So, but beside it, you are talking about what? The two sisters, all right, which are the sarcoplasmic reticulum. Now, remember, this is a muscle what? Fiber. This is a muscle fiber. So, this group of things here is what? The myofibrils. This is the myofibrils. This is myofibrils. I told you the myofibrils make up what? The myofiber. That's the muscle fiber. Now, the myofibrils contains what? Myofilaments. So, if you want to create it from the smallest to the largest, you say actin, myosin, and myofilaments. They make up the myofibrils. The myofibrils make up the myofiber. All right. The myofiber make up the fascicle. Then the fascicle make up the muscle tissue. You go like that. That's if you even want to write anything down, that's what you should write down. All right. So this helps for the contraction and relaxation. I'm coming to this. Now, I have to fly. Now we have elastic filament we call the titin. I'll talk to you about it in a few. Now, jumping all these things, I am coming. Okay, now you must also know dystrophin. Dystrophin is very clinically important. They will tell you where is dystrophin found. Dystrophin, this is the dystrophin. You see, it's connected. It's like connecting this muscle fiber to the plasma membrane. All right, that's what dystrophin do. It's a linker. And if there's a problem with this dystrophin, very, very important, it leads to a genetic problem, leads to what? Something we call a muscular dystrophy, all right? If you look at this diagram, you see different muscular dystrophies, but I'm not here going to talk about muscular dystrophy because I'm not discussing pathology. I'm discussing what? Anatomy and physiology. I remember anatomy is what? Location, or structure. And physiology is function. You need to know the normal for you to be able to know the abnormal. As I keep telling you, as always, I am talking. My mouth is moving, right? That is physiology. And when I am doing like this, my mouth is moving, but you cannot hear me. Oh, you say, oh, professor, something is wrong. Check your microphone. It becomes a pathology. Maybe your microphone is not working properly. That's pathology. Once something is not working properly, it's pathology. All right. Maybe the problem is not my microphone, the problem is my voice. Still pathology. All right. So I'm not discussing muscular dystrophy. Look it up later, or when you get to pathology, or when I discuss pathology, you learn more about it. Now, this is what you must learn. This is something you can't do without. All right. They'll give you this diagram. They will put A, B, C, D, this, that. All right. You need to know what you are looking at. Now, this is a Z disc. Start with this Z disc. This is another Z disc. In between one Z disc and another Z disc is something we call sarcomere. This is sarcomere. In between one Z disc and another. So if you see the arrow, that this arrow is pointing from this Z disc and this arrow stretches to another Z disc, it is called sarcomere. That is baseline. Now, what is the function of this Z disc? This Z disc is holding, or it's a pillar that the actin is attached to. So if you look at this diagram very well, I don't know if you can see it from where you are, you see something like an orange stuff, right? That is attaching to this guy. Another orange stuff that's attaching to this guy. That orange stuff in between the two greens is called what? Actin. Is the tin filament. The two green things are called the titin. That's the elastic filament that is helping to hold the actin properly to the Z disc. Now, this side you see the actin, this side you see the actin. The place where you see the actin alone, so from here to here is the I band. Now you see this big one inside, this purple one inside, that is the myosin, that's the thick filament. Now you see the thick filament extends from here to here, and it is overlapping the actin here and overlapping the actin here. So the part where the thick filament is overlapping the actin, so from here all the way to this place is known as what? A band. Another word for A band is what? Dark band. So this I band is the light band, A band is the dark band. And the alternating I and A band is what leads to what? Striations. If you remember, it's equation. Now, there is a part of this thick filament where it's not overlapped by I band. So this place where you see only the thick filament, it is called what? The H band. And this H band in the middle of it is called what? The M line. That is it. Check it now. Check it now. Is it too much? No. Just learn this thing so that you don't feel it. Okay. Okay, so this place explains all those things we just discussed, but I'm not talking about. Also, the same thing with this. This is also the same thing, all right? If you look at it, what do you see? You see this place that is light. Simple. It's I band. It's light band. It's light. You see this place that is dark. Simple. It's A band. It's dark band. Now, in the middle of this A band, you see a place that is lighter. That's the place you can see only the thick band. That's the H band. Simple. In between it is the M line. And this is, you see, this Z disc in between the I band. This is another Z disc. So from this Z disc to another Z disc is what? Sarcomere. Chicken. Otito. Now you are good to go. All right? So if you understand this explanation, click this button. The button that did this by the, I don't know where it is. Just click it. Okay. So that I'll be sure you understand. Okay, so I'm jumping every other thing now. Now the innervation, we have the innervation atrophy, all right? That's a shrinkage of paralyzed muscle when connection not restored, all right? Skeletal muscle never contracts unless stimulated by a nerve. Always, it must be stimulated by a nerve. They will ask you this question. Okay. Now, we have the somatic motor neuron and we have the somatic motor fibers, all right? Somatic motor neuron is nerve cells, all right? Somatic motor fibers are axons that lead to the skeletal muscle. So you should also remember this. Okay. Okay, yes. So there's something we call motor units. Motor unit is one motor nerve innervating several motor fibers or several muscle fibers. So this is one nerve innervating several muscle fibers. So if it's innervating like 5 muscle fibers plus the one nerve, all of them is called what? Motor units. All right, motor units. So there are some that has small motor units, some has large motor units. Like this muscle in your back of your leg, that's the gastrocnemius muscle. It has almost 1,000 muscle fibers. But this 1,000 muscle fibers is being controlled by what? One unit, one nerve. But we call it large motor units, all right? Then there are small motor units and there are intermediates also. Now, that leads us to the neuromuscular junction, all right? There's something we call synapse. Synapse is where a nerve fiber meets its target. It can be nerve to nerve. We call it nerve to nerve synapse or neuronal junction. It can be nerve meeting a muscle, all right, muscle fiber. That's what we call what? Neuromuscular what? Junction. It's as simple as that. Okay. I want to take my time to explain this now briefly before I fly. Let me see. Okay, so you have something we call synaptic knob. This part that is swollen is what we call synaptic knob. We have synaptic cleft. Cleft is this junction between this nerve and the muscle. This is the muscle here. You see that striation that we saw now. This is the muscle. So this is the plasma membrane of that muscle. Sorry. So a space in between the two of them is known as what? Synaptic what? Cleft. Then we have the Schwann cell. We have the now. These are vesicles inside. These vesicles burst by exocytosis and they have to be activated. All right. So you see this nerve, action potential comes this nerve. Let me see if there's a picture I can use to explain all these things and we move. I don't have so much time. Okay, perfect. So you see, this is the arrow. You see this arrow, action potential is coming down from the nerve. This action potential activates the calcium. Calcium enters this place, bursts these vesicles, pours them into this cleft through exocytosis. You see, it pours it into the cleft through exocytosis. And what happens? These things that it pours into it, it's called what? The neurotransmitters. Specifically for the muscle and the nerve, it's called ACH, acetylcholine. This acetylcholine attaches to the receptors, and immediately attaches to the receptors, it does what? See its attachment. It triggers the ion pump. The ion pump or the ion channel. You see the ion channel? This is the plasma membrane of the muscle. All right. So what happens? Sodium enters inside, potassium jumps out. Sodium enters, leading to positivity of the cell. Potassium goes out, leading to negativity. Sodium enters, leading to positivity, positivity plus 75. Potassium leaves, leading to minus 90. And before you know it, action potential what is generated. That's all you need to know. You understand?